This immersive video will introduce you to the role of clinical laboratory technician working at Illumina at the Wellcome Genome campus near Cambridge in the UK. Illumina are a global genomics company known for leading the market in genome sequences. Their mission is to improve human health. Illumina were involved in the historic project to sequence the entire human genome. One of Illumina's teams is called ILS, standing for Illumina Lab Services. The team sequences DNA of NHS patients with rare genetic diseases or cancer to help with diagnosis and personalised treatment plans. This is the Ogilvy building, where Illumina ILS team are located. Let's take a look inside and meet some of the people who work here.
Hi everyone, my name is Rosa. I'm a clinical lab technician at Illumina Lab Services, and my job consists in performing all the lab tasks that are involved to convert a DNA sample into sequencing results that could be used for diagnosis. Just to show you what we receive in the lab, this is what we call a MIDI plate. It's basically a 96 well, you can see that there's like 96 different spots in there, 96 well plate that holds the DNA of up to 96 different people, and each sample is just extracted DNA that can come from many different tissues. It can come from blood, saliva, it can come from cord in case of babies. So once this DNA is extracted, it is plated in these MIDI plates, and this is what we receive in the lab. So we receive these plates frozen, and the first thing that we do as soon as we receive them in the lab is check that they are still frozen, that they have not been damaged in transit, and that basically they are labeled with their unique identifiers. We do not know anything about the patients that are held in this MIDI plate. We only know barcodes, so all the information is confidential. After we check that the samples are in good state to be processed, that is where the sample journey and processing actually begins, and it happens on the other side of the wall. So yeah, just follow me. When we receive the samples, the first step that we need to take in the lab is checking that these samples have the right DNA concentration in them. So basically that there is DNA in these plates. For that, we basically mix it with fluorescent dye. You can see it in here. It's orange. It's not very fluorescent, but if it attaches to the DNA, actually emits fluorescence. So to measure the fluorescence, we use this piece of equipment. It's an Spectromax. It's a fluorometer. Basically, what it reads is the amount of fluorescence in each specific position. The more fluorescence you've got, the more DNA concentration you have. So we just take the samples from the DNA plate, we move it to this funky plate, which is called fluorotrack, and when the samples are in this plate, it's mixed with the fluorescent dye. So once the DNA and the fluorescent dye are mixed together, we place it in the machine, and then that gives you fluorescence readings, fluorescence measurements. You can see that in every piece of equipment that we've got around the lab, we've got this card system, which shows 4 different states. Up is that it can be used by us. Eng means that this piece of equipment is in engineering. PM means that it needs to be calibrated or have its monthly maintenance check. And then there is another one that is VAL, which means that this equipment is being used by another team. This kind of like gives you the idea of the fact that we are a production production team. We basically just process samples, but we work very closely with so many other teams, like equipment team. Basically, there is a dedicated team of people that just checks that the state of all the equipment that we use in the lab is up to standard. So we work with an equipment team. As I said, we work with a validation team. So basically, they try and make all the steps that we take in the lab faster, more reliable, cheaper. So they basically use the same equipment as we do, but when they're using it, they need to say that the piece of equipment is being used by the validation team, by the developer. So after we've checked that the DNA concentration of the samples that are in the plate are okay, are within requirements, the next step is for us to perform something that is called library prep. Library prep is basically, library preparation is basically transforming these DNA samples in something that the sequencer can read afterwards, because samples as they are cannot be just loaded into a sequencer. They need some processing first. So the first step is transferring these DNA samples that we've received into something that is called a CFP. It's just a metallic rack, again with 96 different positions, and this is done automatically, like by a machine, I mean, and you can see those machines right at the back. So we've got 5 of them, and they are liquid handling robots, and what they're going to do is just transferring the samples from the plates that we receive into these very cool metallic racks. Why? Because the first step of this library preparation is fragmenting these DNA samples into very short fragments, millions of them. And how do we fragment those DNA samples? Using this instrument right here. They are basically sonicators, and they work with some wavelengths that basically fragment the DNA to very short fragments. It's very similar to when you see in the movies someone singing very high pitched and breaking the glass. This is going to sing high pitch and it's going to break the DNA into smaller fragments. So if you come with me, we're just going to load this rack in the instrument. We just click run, and as you can see, the plate is then submerged underwater to improve the travel of these wavelengths. And you cannot hear it because it's very low, but if you were to put your ear close to it, you could hear the small like sound. So yeah, it's basically just shearing the samples, and you can see that it's doing it column by column, so it's going to do the whole length of the plate. I just want to introduce to you another very important part of the lab, which is our Hamiltons. They're liquid handling robots, and as you can see, we've got 5 in this lab. Basically, what they do is they automate the processes, so we can process way more samples than we manually could. And they do a lot of fancy stuff. They move samples across different positions in the tape. They can incubate plates because they've got heaters, they've got shakers. It's a really cool piece of equipment to work with. So at the moment you can see that the Hamilton is transferring the samples from this metallic plate at the front. This is called a CFP plate. Is basically contains the fragmented DNA, and it's just moving that fragmented DNA to the plate at the back. So as you can see, it goes into the first plate, and then using these channels, so it's just a channel of tapes, it moves it to the plate right at the back. It not only has channels, it has like what is called a core head, so it's a 96 fixed tip, like tips, so it basically can move 96 samples at the same time in a block instead of doing it 8 by 8, which is limit now. So after library preparation is finished, we have our prepared DNA ready to be sequenced. It has been fragmented. We've added those little extra pieces that the sequencer needs to read the DNA sequence. So is the prepared DNA what we've got in here, and we have to load it into the sequencer in the lab next door, which we're going to go into now. And we move it through this hatch. And I'll see you in the other lab. Hi again. You'll notice that we've got different color lab coats in this lab. This is called post-PCR lab. The one that we've just been in is pre-PCR lab. And basically we have two separate spaces to reduce the potential for contamination, so it can be contained in one space and not spread across the whole pipeline. So that is why we've got two different labs and two different color lab codes. And if you follow me in, you will see how we load the samples into the sequencer. So we are in post-PCR now. You will notice that it's a little bit noisier than pre-PCR. That is because we've got 12 sequences in the background. They can be quite noisy because the ventilation system is quite strong while they're working. So yeah, it's a little bit noisier in post-PCR. Once we've got our prepared DNA, the library preparation, the first step that we take in this lab is actually checking that everything has gone well during library preparation. And for that we use this piece of equipment. It's a light cycler, and basically it performs a technique that is called qPCR, in which basically is going to measure the DNA concentration, basically to assess that there is DNA concentration in this new plate, and basically we haven't lost DNA within the process, and everything is good to be loaded. So this is the first step that we perform in post-PCR. So once we've checked that our library preparation is okay, that it's got DNA in it, the next step that we take in post-PCR is loading these samples into what is called a flow cell. This is what it looks like. It's basically a glass slide, and as you can see, the Hamilton has like a channel of tips. So those channels go into those very tiny holes, and it just dispenses the sample into this glass slide, and it's called a flow cell basically because when it dispenses it from here, it just flows across the glass slide to the other end. If we were to zoom in this glass slide, you would see that it's covered by millions of micropores, and those micropores are where the DNA attaches to in the flow cell. So once the sample is loaded in this flow cell, we put it in inside of a sequencer. You can clearly see that it's got a deck. At the moment it's open, so we just position this flow cell in the deck, and then we just close it, and the sequencing begins in the Nova cell. That was the tour around the lab. I hope you enjoyed it. I would like to say that I personally recommend working as a clinical lab technician because it's a job that basically involves a lot of scientific knowledge, but also requires a lot of troubleshooting and thinking on your feet, and sometimes being creative with solutions. So yeah, it's a job that is different every day, and also it helps you develop skills not only in the lab environment, but also because we work very closely with bioinformatics, with research teams, with engineering teams. You can always learn a lot of different skills related to those teams, and Illumina really promotes that career growth. So yeah, if you like science and like an active kind of job, definitely I would recommend this position. Just so you have a little bit of background of what me and my colleagues did to get to this position, we all have undergraduate bachelor's degrees in a relevant scientific field, either biology or chemistry. We've got biochemists, people that have studied natural sciences, biomedical sciences, so yeah, if that's something that you're interested in pursuing, then this could be the right place for you after you finish uni.